All right, so sympathetic nervous system, we're going to finish it out. We talked about the thoracolumbar exit for our spinal nerves. We talked about our long preganglionics. We talked about our collateral ganglia. Now we have to talk about an honorary member of the sympathetic nervous system. This involves our adrenal glands. So we talk about the sympathoadrenal system. Now, you're probably familiar already with the name of the hormone that floods through your body when you get really angry or really afraid. What is that hormone? Adrenaline. It's called adrenaline because it's produced in the adrenal gland, and specifically in this middle part called the medulla of the adrenal gland. And this is where we find these cells called chromaffin cells. Now, another name for adrenaline is epinephrine. And principally what chromaffin cells produce is 85% of their secretory products is epinephrine, 15% is the neurotransmitter norepinephrine. And it's because they do make some of this neurotransmitter and the fact that they are stimulated by preganglionic neurons, then these are sort of honorary postganglionic neuron members, even though they're not really neurons. But here's the big thing I want you to remember. And I hammer this into my pre-med students. We've talked about it a couple of times. Epinephrine, aka adrenaline, is not a neurotransmitter. It's a hormone. Write that out beside it. That's one of my pet peeves. If someone says epinephrine is a neurotransmitter, I want to give them a 0 for the whole test. Epinephrine's a hormone because epinephrine secreted by these chromaffin cells goes into your bloodstream. It doesn't go into a synaptic cleft, much like our neurotransmitters do. Now, one of the advantages of going into your bloodstream is epinephrine can spread throughout your entire body almost instantaneously. Because we talked about being at the red light, light turns green, you start to pull out, and someone runs the red light right in front of you. How long does it take for your body to wake up? That fast, because adrenaline flooded your bloodstream. Adrenaline, the hormone, also called epinephrine. So that's this last part of our sympathetic nervous system. Sympathetic is what or what? What do we call it? Fight or flight. Now we're going to jump to the other one. And remember, I said that the parasympathetic was largely going to be opposite of the sympathetic. So while sympathetic was fight or flight, parasympathetic is rest and digest. Really active for sympathetic, very inactive for our parasympathetic. Now, in our big picture that had the red and the blues for sympathetic, parasympathetic, we saw that the spinal cord exit for the sympathetic system was in the middle. Do you remember those numbers? T1, where did we go down to? L2. Thoracolumbar, that was our sympathetic. Our parasympathetic is going to be craniosacral. So it's opposite in the sense it's not in the middle, it's at the top and the bottom. And we'll come back to this. It'll be on the next slide as well. But I want you to see, here's our big picture. There's our thoracolumbar, but there is our cranial, because we're going to have cranial nerves that are going out in the blue. That's our parasympathetic, as well as our sacral nerves that are going to exit and bundle together into the pelvics to innervate the lower part of our body. So, which nerves? We have 4 cranial nerves. We actually have 12 altogether. We're going to talk about these 4 as part of the parasympathetic nervous system. Starting at the top and going down, we're going to start with number 3. And on the exam, if you put a number or a name, doesn't matter to me, just get the right one in the right place. So cranial nerve number 3, called the oculomotor nerve, is a pure motor nerve. There are no sensory fibers going back to the brain in it. And the oculomotor nerve is going to control the size of the pupil by controlling the contraction state of the iris. the color part of your eye. You're doing that in lab right now, right? The eye. Now we're going to see in a little bit the physiological responses that happen in a sympathetic versus parasympathetic, and the size of the pupil is going to be one of those things. The oculomotor also controls the contraction state of the ciliary muscle. The ciliary muscle controls the size and the shape of what? The lens of the eye. And that's for focal accommodation, distant versus near vision. That's all regulated by cranial nerve 3, oculomotor. Now, cranial nerve 7 is the facial nerve. The B stands for both sensory and motor information traveling in that nerve. And when you think about what's going on in the face, it's not surprising. You would see activation of your mucus glands in your nasal sinuses. Lacrimal glands, what are those? What do they make? Tears. And then your salivary glands. Those are all regulated by cranial nerve 7. Cranial nerve 9. This is a big word. Glossopharyngeal. Have you ever heard of glossa before? Prefix glossa. Glossa means tongue. So glossa, tongue, pharynx, back of your throat. This is what's going on in the mouth. It is both sensory and motor. And it's largely going to innervate your largest salivary gland called the parotid gland. Do you know where the parotid gland is? You'll get that, I'm sure, in digestive in 239. But it's back sort of at the hinge point of your mandible. It's the one when a kid has the mumps that it swells up. But even though it's the largest of the salivary glands, it doesn't produce the bulk of the saliva, which is kind of weird. But that's going to be innervated and controlled by cranial nerve 9. Cranial nerve 10, the vagus, sensory and motor. Vagus in Latin means wanderer. You see why they named this one the vagus? Where does it go? Practically everywhere in the abdomen and the thorax. Heart, lungs, GI tract, liver, pancreas. So those are your 4 cranial nerves as part of the parasympathetic division. Then S2 to S4, down in our sacrals, our pelvic nerves are going to the lower part of our digestive tract, rectum, bladder, reproductive organs. So really these cranials that start at the top, they branch and go everywhere except for the lowest parts of your body, which are regulated by these pelvic nerves. And again, we can see those pelvic nerves illustrated here going to uterus. There's our bladder, lower part of the GI tract. Here are our 4 cranial nerves, and here's the vagus, number 10. You can see that it's going everywhere throughout the body, even down low to like the kidneys. Here's some part down to the small intestine, transverse colon. So it goes all over the place. Now I want you to notice something. When we talked about pre versus postganglionic in the sympathetic, which was the longest? In the sympathetic. The post. See, the pre's, the solid lines, they're fairly close here to our chain of ganglia. Some go a little bit further. And with parasympathetic being opposite, which is going to be the long. The pre. Notice these solid blue lines. They extend quite a distance away from the spinal cord. The vagus even actually goes practically out to the tissues themselves before it forms a synapse. But our 3 cranial nerves here at the top, they're going to form a synapse in ganglia that are fairly close to our final organs in the head. And those are going to be called the terminal ganglia. And just like we have 4 cranial nerves, we have 4 terminal ganglia. But don't get tripped up because only 3 of those cranial nerves go to a ganglia. The vagus doesn't. And so if you notice here in the middle, the facial nerve has 2 terminal ganglia that it actually will form synapses in. So you can think of this like a double stuffed Oreo. There's these 2 in the middle. So here are the names of our terminal ganglia, and the top and the bottom, it's actually nice because they go to and are named after the organs that are being innervated. The ciliary ganglia, which is innervated by the oculomotor nerve, it's going to innervate the ciliary muscle. The parotid ganglia, innervated by the glossopharyngeal, it's going to go to which salivary gland? The parotid. So those just link up with where they're going. These two in the middle with the facial nerve, they're a little different. Because while the submandibular, that is the name of one of our salivary glands, and it's actually the one that produces most of your saliva. So even though it's innervated by the facial nerve, this big weird sounding word, pterygopalatine, this is going to be the one from fibers of the facial nerve that will then go on to your nasal mucosa and the nasal glands and your tear glands. So that facial nerve will split into two. And we'll be able to see that in our red and blue picture. We'll see the facial split and go into the two, the parotid and the glossopharyngeal go into the parotid, and then the vagus just goes to the organs without going to one of our terminal ganglia. All right. So, here in our picture, we can see our nerves, three, going to the ciliary, and there's our facial. Do you see how it splits? Goes to the pterygopalatine, the submandibular, and then here we have the glossopharyngeal coming down here to the parotid, our four terminal ganglia, and of course the vagus just going all over the place. So red, sympathetic, blue, para, solid lines, preganglionic, dashed lines, postganglionic. But remember, some of those postganglionics are going to be right in the tissue itself. That's how short those postganglionics are actually going to be. So what does all this hard wiring do for you actually? What are the physiological consequences of dominating a sympathetic versus a parasympathetic? So let's look at the heart. If you are afraid or angry, what is your heart going to do? It's going to speed up and it's going to beat harder. When you're sleeping, what does it do? It slows down. Fight or flight, rest, digest. Now the iris, controlling the size of the pupil. In a fight or flight, what's going to happen to your pupil? It's going to get big. You want as much visual information coming in as possible. Now, I've purposefully put the opposite because I want you to think the opposite. And so the opposite of dilation is constrict. Understand that's not actually accurate. When you're sleeping, your pupil doesn't go down to a pinpoint, okay? Depending on what you took before you went to bed. But it doesn't dilate. Does that make sense? Dilation here, no dilation there. Blood vessels, this is a little bit trickier because in a sympathetic response, most of the blood vessels in your body, especially those going to your internal organs, most of those constrict because you need your blood going to your skeletal muscles. You don't care if it's going to your digestive tract, your reproductive organs. You've got to have more blood going to the the skeletal muscles. So we constrict these in a sympathetic, and even though we say dilation in a few, understand they're not constricted. And this dilation, by and large, is going to happen to your gastrointestinal tract. Because what is this? Rest and what? Rest and digest. That's what's going to be responsible for that dilation. Now, in a sympathetic response, your heartbeat, it speeds up. Your smooth muscle, skeletal muscles, I keep saying smooth. Skeletal muscles continue to prepare to do something active. So how is your respiratory rate? Your demand for oxygen, how is that going to be affected? You're going to need more oxygen, right? So your bronchioles, these airways of your lungs, they're going to open up. And the way they're going to open up is the smooth muscles that wrap these bronchioles are going to relax. And when they relax, that's going to cause the cartilage rings of these structures to rebound and elastically open up those airways. Now, likewise, we've got dilation over here for opposite purposes. We say constrict, but understand when you're asleep, you don't stop breathing. Well, unless you have sleep apnea. But your airway simply doesn't dilate. So if you want to write in here, constrict pupil, here, constrict, just out here, put it, no dilation. All right? But again, the opposite of dilation is constriction, so that's sort of why I put those in there. Now, when we get to your GI tract, your digestive system, things flip. We've been doing a lot of active stuff over here in sympathetic. Well, in your gastrointestinal tract, in a sympathetic response, your stomach, it's closed for business. There's nothing going on in your stomach. No secretion of digestive enzymes at all. And when we talk about the peristalsis that happens to move food through your stomach, through your intestines, man, we got a lot of movement going on in a parasympathetic. Sympathetic, it's shut down. Food's not moving. That's why sometimes, your parents say, oh, don't go swimming until like 15, 30 minutes after you've eaten. If you've got a lot of food on your stomach, and your body's trying to digest that food, and all of a sudden you jump in the pool and you start getting really active, your digestive tract shuts down, and so you're probably going to get indigestion. And that will feel like your stomach, what? Cramping. Don't go swimming. You got 30 minutes after you get a cramp, right? What's your stomach? It's because of what's happening between the parasympathetic and sympathetic. Sphincters. Do we understand sphincters? I know digestive is 239. It's these valves that control the passage of material from one point to the next in your digestive tract. You have something called the Pyloric Sphincter. Controls food from your stomach to your small intestine. You have the ileocolic valve, small intestine to the colon. Well, guess what? They're open for business in the parasympathetic. Sympathetic, they are shut down. Whether you digest that Big Mac or not is... Your body doesn't care when you're running away from a lion. You need all that material going to your skeletal muscles, going to your heart, opening up your lungs so that you can live to eat another Big Mac on another day. Now, here's a chart that sort of compares. You don't have to memorize this chart. And in fact, it's pretty logical if I ask you about a physiological response, sympathetic versus parasympathetic. Just think, what has my body got to do to be active? To win a fight or to run away from something? Versus what is my body likely going to be doing when I'm sleeping? And that's how you can link those two divisions up as far as the physiological effects that are going on. All right, now we're going to shift gears a little bit as we come to the home stretch. The home stretch, that's the toughest part of the race, right? But that's the part where you gotta give that last little bit of effort to cross that finish line. We're getting close. But now we're going to talk about the neurotransmitters and the receptors that are involved in making these physiological effects happen. So first, we're going to talk about cholinergic neurons. Now, when I say cholinergic neurons, what neurotransmitter is produced by these neurons? Do we have a neurotransmitter that's got choline in the name? Acetylcholine. That's how we get this name. So all preganglionic neurons, whether they're sympathetic or parasympathetic, they're all going to produce acetylcholine, and therefore at the synapse between the pre and the post, that's always going to be excitatory. Because it would be stupid for a preganglionic neuron to be inhibitory. I mean, why even start it in the first place? If it's not going to need the signal, just don't even start it. So all preganglionic neurons of both subdivisions are going to secrete acetylcholine. Now, all of the parasympathetic postganglionic neurons are also going to secrete acetylcholine, and most of them, not all, but most of them are also going to be excitatory. We're going to see that the postganglionic parasympathetics that go to the heart are actually going to be inhibitory, because in parasympathetic, what does your heart rate do? Slows down. So you have to inhibit some of those pacemaker cells so you can slow the heart rate. Now, one more little exception. Some of our sympathetic postganglionic neurons, specifically those going to the blood vessels of our muscles and blood vessels to our sweat glands, those are going to be excitatory, and those are going to be cholinergic as well. And again, in this response, if we're talking about sympathetic, we need those smooth muscles of those blood vessels. Most of them going to our visceral organs. We need those to shut down so more can go to our skeletal muscles. Now, here's my sort of synopsis picture. Now, if we look across at our rows, along the top we have the preganglionic neurons. Along the bottom we have the postganglionic neurons. Now, as we look at our columns on the far left, this is our parasympathetic division. And then here's our sympathetic division. Notice we have 3 of them. There are actually 4. They don't illustrate the 4th one, but we're going to talk about them. These 3 divisions are different because of the receptors we're going to add down here at the bottom. But that's coming up next. But can you see the difference between this green color and the blue color down here? Might be hard if you're kind of close to the back. All the green dots that you see are acetylcholine. So all of these are cholinergic, except for those few exceptions we had. But now look at here with the blue, what do we have? Norepinephrine. So we're going to talk about those in just a minute. Now, I'm going to take you back a little bit. Have to think back. I think maybe Unit 2. It's not too far ago. Think about our neuromuscular junction. Motor neuron innervating a skeletal muscle. Okay, are we there? That motor neuron was cholinergic. It produced acetylcholine. Acetylcholine crossed the synaptic cleft and it bound to a receptor in the skeletal muscle. That was a cholinergic receptor. How many acetylcholine molecules had to bind to it? 2. And when 2 acetylcholine molecules bound to that receptor, what happened to that receptor? It opened up. It was a closed ion channel. We're going to give that a name now. It's not something new we're introducing. We just give it a name. That is a nicotinic receptor. That's the name of the one we put in our skeletal muscle membranes. We're also going to see that elsewhere in the human body, but mostly we're going to see it with skeletal muscle. And remember, it's always excitatory. Now, ligand, that word means chemical. In this case, neurotransmitter. So this is a neurotransmitter-gated ion channel. Again, you know what that is. You know what it looks like. We've played with it before. Now we're going to throw a second type of acetylcholine receptor. This one you're going to find on cardiac and smooth muscle, and it is called a muscarinic type. It's only going to bind one acetylcholine molecule, but instead of it being an ion channel, it is a receptor that is going to send a signal to a separate ion channel. And that messenger that's going to go from the receptor to the separate ion channel, they're called G proteins. So in all of our cells, the receptor is going to be the same. The G proteins are going to be the same. But the particular ion channel of that tissue can be different so that the result is either inhibitory or excitatory. So let's just look at this for a second. You remember this one? Muscarinic receptor, 2-acetylcholine, ion channel, sodium, potassium, you get an excitation. But when we look at our muscarinic type receptor, here's the receptor, there are G protein messengers. In the case of the heart, when you bind norepinephrine, acetylcholine in this case, then that's going to send these G proteins over and cause this potassium channel to open. Potassium's high on the inside. If potassium leaves the cell, potassium being positive, if it leaves the cell, what's going to happen to our membrane potential? More positive or more negative? More negative. Hyperpolarization. Is that an excitatory potential or an inhibitory potential? Inhibitory. So it's an IPSP. And we're kind of reviewing some of those things that we've worked with throughout the semester. But that's because the G protein opened up a channel. Now, look in smooth muscle. Over here, still acetylcholine, still the same receptors, still our G proteins. But in the smooth muscle of our digestive tract, for instance, we've got a potassium channel that was open. Well, now we close it. But we still have our sodium channel over here, and sodium continues to come in. Positive ions coming in. Is that an excitatory or inhibitory potential? Excitatory. So you see how you can have the same stuff but different ion channels, and it can be either inhibitory or excitatory? So that's how we work, and that's how the difference we have between our nicotinic and our muscarinic receptors. Now, keep this in mind, the receptor and the G proteins, because we're going to see it again in just a few minutes. Because now we talk about the adrenergic types of receptors. Now, cholinergic receptors, what was that neurotransmitter? Acetylcholine, choline in both of those terms. This one is really tricky. Because guess what neurotransmitter is produced in an adrenergic neuron? Norepinephrine. That's how in the world did they come up with this? Follow my logic, okay? Norepinephrine. That name sounds a lot like a hormone called epinephrine, right? Norepinephrine, epinephrine. What's another word for epinephrine? Adrenaline. Where is adrenaline produced? In the adrenal gland. Get it? Adrenal, adrenergic. You only have like 15% of those chromaffin cells producing norepinephrine, but I guess adrenergic just is easier to say than norepinephrinergic, and maybe that's why they arrived at it. So, adrenergic neurons. Most of our postganglionic sympathetic neurons are going to produce norepinephrine. And again, like we've seen before, this is going to be facilitated through a receptor and G protein, so it can be either excitatory or inhibitory. So in the heart, norepinephrine is going to cause an increase in your heart rate. But in the smooth muscles of our airways, those are going to be inhibited by norepinephrine. And if you inhibit those smooth muscles, that's going to allow the bronchioles to open up so you can get more oxygen into the lungs and therefore into your blood. Oops, let me go back. Everybody got that? So our next picture, again, is just illustrating. I mean, this looks just like what we had for our muscarinic acetylcholine, but now norepinephrine, receptor, G proteins. Those signalers go over and activate other proteins in the cell surface. That's going to end by activating an ion channel, either opening it or closing it. Depending on the ion channel, depending on the tissue, it's either going to be excitatory or inhibitory, much like we saw with our muscarinic acetylcholine. Now, what's going to make part of that difference is the receptor itself. Because we had two for acetylcholine, right? Nicotinic, skeletal muscle. And what's the one on smooth and cardiac? What's its name? We just learned it. Muscarinic. Well, our adrenergic receptors, there are 4 of them. 2 alphas and 2 betas. But notice, all signal via G proteins. The only ion channel we're dealing with is that nicotinic skeletal muscle receptor. Everybody else is G protein. And that nicotinic is the only one that can be purely excitatory. So our alpha-1 receptor, this is going to be present on vascular smooth muscle. What is the term vascular? What does that mean? Blood vessels. Blood vessels. So alpha-1, that's in the smooth muscle of our blood vessels. And that's going to cause the constriction of those blood vessels, so it's going to shut down blood flow. Alpha-2 types of adrenergic receptors. We see these in many ways on our presynaptic membranes. which in a sense is going to be a negative feedback. Do we remember negative feedback? It's a way of shutting things down, keeping it from getting out of control. Because a sympathetic response is almost a positive feedback chain reaction. And your body can't keep doing that. It can't getting more and more angry or more afraid. And you've got to be able to stop that response at some point. So this whole idea of a negative feedback for the sympathetic system is great because we have a lot of alpha-2s in the brain. And when you have a sympathetic response and you have all this norepinephrine, this alpha-2 helps to shut it down or to keep it from getting out of control. So for our alpha-2s, I want you to remember, present in the brain, and it's a part of a sympathetic negative feedback mechanism. So that we don't have to feel that way for days and weeks. Because sometimes as fast as it starts, it kind of goes away. Now our betas, beta 1 and beta 2, are basically tissue specific. So beta 1 in the heart, that's going to cause our heart to beat faster. Whereas beta 2 that you find especially in that smooth muscle of your bronchioles, it's inhibitory because we want those to open wide up. So again, like we saw in our muscarinic, we've got differences going on between what's happening in the heart versus the smooth muscle of our airway. Because in a parasympathetic, we need the heart to relax, and we don't necessarily need the bronchioles to constrict, we just don't need them to dilate. Versus a sympathetic response, we need to go faster, and we need to relax this to open that up. And so basically, you can summarize all of this in our picture, except for the alpha-2, which is in the brain. They don't put this in our picture. But here in our picture, now we put in our cholinergic receptors. They've got the muscarinic listed here. But there's our alpha-1, vasoconstriction of the viscera and the skin. Beta-1, increased heart. Beta-2, dilation of the bronchioles of the lungs. So that's why we have these 3 columns. Just understand that fourth goes to your alpha-2's negative feedback in the brain.